Welcome to the short case study where we want to look at how we can use the ReadStore database to power a single-cell RNA-Seq analysis in R using the Seurat package. Seurat is a widely used library for performing single-cell RNA-Seq analysis, and what we want to take a look at is how we can make the access to individual projects and datasets and intermediate files easier using the ReadStore database. And for this, we will look at a dataset that contains of 17 single-cell RNA-Seq libraries from a sting-associated vasculopathy with onset in infancy. So this is an autoimmune disease from a publication that came out last year in Cell Reports Medicine. And what we want to do now is load these datasets using the ReadStore database. So we have registered the datasets there already, merge the datasets for single-cell RNA-Seq analysis, load in the metadata from the ReadStore database, and then combine the Seurat objects and store some of the intermediate files again in our ReadStore database. So we're now in our ReadStore web application where we can get an overview of all the projects and datasets. So you can run this web app simply in your local server or on your private cloud accounts. And when you log in, you get this user interface. And here we see that in the projects, we have already created a project for this study. And if we move to the dataset section, we get an overview of all the single-cell RNA-Seq datasets that are registered here. If we want to get more information on a specific project, we can simply filter out individual projects. And since we only see this rather cryptic identifier here, we can also toggle on the metadata view. And this helps us to get further information on each of these datasets, for instance, the disease group or the treatment status here that will later on be important for a single-cell RNA-Seq analysis. If we want to get more information on an individual dataset, we can just select it here and then move down to the detail overview. And here in the Pro Data, Process Data tab, we see that we have one count matrix in H5 format attached to this dataset. And this we can later read out using the R SDK to pull these data directly from the database. So we're now in our RStudio environment where we can explore the datasets and where we will see how we can structure a workflow around the ReadStore package. So first, what is happening here in the first parts is that we are installing ReadStore via the remotes package directly from GitHub. And then you can import the ReadStore package here. And the first operation is to create a client for our analysis here that connects to the database. The next step is that we want to get a list of all the projects. For this, we can just run the list projects over operation, and this will give us a data frame that contains all the projects, which we can view here in the data frame viewer. And here we see that we have, in fact, only one project, as we saw in our app. That contains certain metadata, attachments, and an ID. And what we will do next is to get explicitly the metadata from this project. And for this, there's another command, list projects metadata, which helps you to return all the projects in a data frame. And this data frame contains here two metadata entries for this project, which is the tissue and the cell type. This is very useful because you can store your data very flexibly in the ReadStore database, and then for your analysis you can always retrieve this data frame and you have all your project data right away structured. So next we will list all datasets that are part of this project, and for this we use the list datasets operation, and here we can provide a project name. And we take this SAVI study here, and that should give us all the datasets that are associated with this project. We can view these datasets also in a data frame viewer. And here we then get a similar overview that we received in our ReadStore app. And same here, we can also perform a metadata query. And this gives us then the overview of all the dataset metadata. Here we're adding the original dataset name in, and if we go to the data frame viewer, we can get an overview here of all the metadata variables that we would need for an analysis for each of the data sets here. And that's very useful because we don't need to import anything from Excel files or from somewhere. We have everything stored in this simple database. And what we need for our analysis is actually the count matrices for the single cell RNA-Seq, and those are stored as process data. And these process data can be listed for each of the data sets with the list pro data command here. And we provide a project name, which is the SAVI study again, and a data type. So here we can set specific data types for entries here, and this will only now return all the entries that are count matrices in H5 format. So we're free to choose this for any type of process data we would want to store in ReadStore, and with this we can retrieve these process data. And if we take a look at our viewer, we can see that here those now contain, for instance, the count matrix data type. These entries are also versioned, and they also contain the data sets that they are associated with, along with the file path where this data set can be found. Those process data are now the input for our single cell analysis. So here we are going to perform some operations on each entry of this data frame. I put this here in this for loop. That's of course not the best way of doing it in R, but for the sake of illustrating this, we're going to loop through each entry of this data frame individually. What we're doing here is we extract the dataset name and the path for each of the entry, and then we create a Seurat object. So we're loading the count matrices here in this raw data entry using Seurat, and then we create the Seurat object itself. So the Seurat object is the basis for single cell analysis in R. And here we already perform a filtering step. And all of these Seurat objects we're then allocating in a list because we want to run a second processing step here. What this gives us here, if we take a look at this list, is a list of all the 17 Seurat objects that have been filtered and that are now contained here. And in the second step, we want to actually upload these Seurat objects, save them and upload them to our ReadStore database, because if we want to repeat this analysis or run the analysis in a slightly different way, then we don't want to run this pre-processing again, but we want to have access to the Seurat objects directly. So here in this second for loop, we're going over the individual Seurat objects in this list, and we're running the save Seurat RDS command. And this will save the individual Seurat objects that we created now to a certain file location. And then in a second step, we can upload these process data, which is now our Seurat objects, into the ReadStore database, so that the next time we want to run a similar analysis, we can directly go to the database and get the file path to the Seurat objects from the database here. So for this, we need to define a name. In this case, we call it Seurat RDS. We need to define the file path, which is the location where we stored this object, the data type. We can store some meta information, for instance, how we filtered the data sets here. And of course, we need to assign those data sets back to the original data set. After we run this operation, we can again list all the process data. This time we are filtering this for the data type Seurat, which we just created here. And that should give us now an overview of all the Seurat objects that we just uploaded. In our viewer, we see that we have now the corresponding number of Seurat objects here, along with the data sets and the location where the objects are stored. And that's of course helpful, because if we want to run this analysis again, we can just go to our ReadStore database and pull all the file path plus all the relevant annotations from this database. And this facilitates our analysis because we don't need to search for our files anymore. And then finally, we would go into the actual single cell analysis. We're not going to perform a full analysis here, just show the first steps. And then for the rest, there are a lot of great tutorials. So here we are now, for instance, extracting the cell IDs, which are the dataset identifiers, and we're merging all those 17 Seurat objects here, which would give us a big integrated matrix. And then we can perform normalizations on this and find variable genes and all the other steps that are relevant for a downstream single cell analysis. But again, the point is really that we can manage all our metadata and pull all relevant data sets from the ReadStore database. And with this, we can make our single cell analysis very reproducible. We can scale it very easily, and we can also repurpose now our scripts in a very lean way, because we have the same interfaces for accessing data and metadata and process data using the ReadStore database. And if you have questions or comments on this, please leave them here. Otherwise, you can find a demo of ReadStore on our website, which I'm going to link. And otherwise, I wish you a great day.